So I welcome you all in the next video tutorial. So the next plant we are having with us is the Taxus. Yes, you are seeing the photograph of the Taxus plant. In Sanskrit, this plant is known as the Talis Patra, and there are different varieties of Taxus plant available. So Taxus consists of leaves, bark, and roots of various species of Taxus belonging to family Taxaceae. And majorly there are four prominent species are used, that is Taxus baccata, Taxus brevifolia, Taxus canadensis, and Taxus cuspidate, and they are known by the different name. For example, we can say about the this plant is basically known as the Himalayan yew plant. So this is the Himalayan yew. So for example, English yew is known as the Taxus baccata, whereas the Pacific yew is the Taxus brevifolia. Again, the American yew is the Taxus canadensis, and the Japanese yew is the Taxus cuspidate. This is the bark part of the yew plant, that is a Himalayan yew or the Taxus species. Again, these are the bark. So yes, this is very beautiful fruit of the Taxus. These all are the photograph related to the fruit. So about the habitat, so it is a very slow growing evergreen gymnospermous tree, just like your Devdar. It is a gymnospermous tree found in India, Canada, America, and also in the Himalayan region up to an altitude of 2000 to 3500 meters. So about the chemical composition, see this complex, very complex structure. This is the structure of the very important compound that is present in this plant is the Taxol. So this is the Taxol compound. So the main compound, that is the Taxol, is present in all parts of the plant, especially in the leaves, root, and the bark. But the composition varies from the different plant parts. So these taxanes are the class of diterpenes, and they were originally identified from plants of the genus Taxus, that is the yews. And it has a feature of taxadiene core. So basically it is a diterpene with a taxadiene core. And the two form, that is the paclitaxel and the docetaxel. These are widely used as a chemotherapy agent, that is in the use as a anti-cancer drugs. See again, taxadiene is a diterpene, okay, and it is the first committed intermediate in the synthesis of Taxol. So this is very important compound. Just see here the taxadiene nucleus. So this is the taxadiene nucleus. Okay, that is the taxa 411 diene. And in the structure of Taxol, this is the taxadiene nucleus. So just see, the Taxol was first isolated by Wall and Vanni in the dried bark, that is only 0.02%. And this is the basic taxoid ring system. You have to remember this structure. This is the basic taxoid ring system. So in different species, that is the Taxus baccata, Taxus brevifolia, etc., the concentration of Taxus is very low. That is, it varies from 0.007% to 0.01%. So you can imagine it is present in very minute traces. And presently it is obtained from the stem bark of the Taxus brevifolia plant. And there is another problem, that this plant is very slow growing plant. So just imagine, it needs at least 60 years old 3 to 4 trees to get 1 gram of Taxol. So you can imagine only 1 gram of Taxol is produced from 60-year-old 3 to 4 trees. So it has a very low concentration. And 50 to 150 milligram of Taxol are obtained from 1 kg dried bark. So only 50 to 150 milligram of Taxols are obtained from 1 kg drug. So scientists are working on different alternative method to obtain Taxol in the laboratory, that is through the biotechnology or any other method. Actually, there is a compound that is present in the leaf, that is the 10-D-acetyl becatin 3. This one is the compound, that is 10-D-acetyl becatin 3. So this compound is present in the leaf part, and it has been reported that leaf contain this compound, that is a 10-D-acetyl becatin 3, which can be comparatively easily converted to Taxol. So this is now been used as a alternative for the Taxol production, because we can easily obtain leaves also. Leaves are also present in the larger amount, and it is very easy to collect the leaf part also. And leaves are also very quickly regenerated, or they can be harvested easily. So see again the structure of Taxol. This is another important part of the Taxol. This ring is known as the oxetane ring. Just see the structure of oxetane nucleus. So this is the important ring, because oxetane ring increases this activity of this compound, whole compound, that is a Taxol. Look at this. This is the oxetane ring. So let's see the mechanism of action of Taxol. So here you can see it prevents the mitosis and eventually led to the cell death. So it is used as an anti-cancer, and we know cell is the uncontrolled growth of cell, so it will stop the uncontrolled growth by preventing the mitosis. So let's see the mechanism of action of Taxol. So the biological target for Taxol is microtubules, that is produced from alpha and beta tubulin proteins, and these microtubules are responsible for the formation of the mitotic spindle, which is necessary for the further cell division. What happened? Taxol bring about the polymerization of microtubules, due to which the microtubule formulation, or we can say the microtubule formation, is much enhanced, which causes detrimental effect on dividing cells and leads to the blockage of the cell cycle. See again the microtubules. So basically these are the log chain of protein, that is the tubulin, and they play a key role in mitosis. As we know, Taxol is a mitotic inhibitor. It acts in the mitosis phase. So what happens is microtubule, they serve to give a cell shape and aid in cellular motility and play a very crucial role in cell division. What is happening? Taxanes or the Taxol, they stabilize microtubules, because the drug binds to the microtubules and prevent their breakdown. Thus, they shift the depolymerization or polymerization process to accumulation of microtubules. Due to this, the microtubule formulation is much enhanced, and which ultimately leads to disruption in the cell cycle pathway and leads to cell death. So before going into much deep, first of all, just clear the concept of microtubule dynamics, because we are focused on the microtubule. So microtubule means dynamics, means it is very unstable, means it refers to coexistence of assembly and disassembly, means both processes are done, assembly and disassembly, for the production of these microtubules. So just see here what is microtubule assembly and disassembly. So at the tip part, microtubule, they assemble also and they change their shape, they disassemble or disassemble also. Look at this. Now how paclitaxel act? So paclitaxel, it act on the beta tubulin protein. It binds to the luminal binding of the microtubules. Here it is. The paclitaxel is acting, and it blocks the disassembly of the microtubules. Okay, here it is. This is the paclitaxel, and this is posed. Means the shrinking is not done. Means the paclitaxel is blocking this microtubule in this shape. Just see the process of mitosis. What happened? Shrinking occur, and it will leads to the telophase. And how paclitaxel is acting? Look at this. It blocks the shrinking of the microtubule and the disassembly of the microtubule here. So it will no further resulted into the cell division. So it blocks the mitosis phase also. And in cell cycle, it acts on the mitosis phase.